In this video, we will discuss about the HIV virus in detail and also the life cycle of the HIV infection and to reach to a point where it is been defined as AIDS, that is acute immunodeficiency syndrome. So let us start with the first figure. First figure shows the HIV virus, and we have understood this in many of the HIV videos, HIV related videos. So let us quickly go through it again for the new viewers. So this GP120 is the envelope protein that helps to form a spike protein, and this green color is known as capsid that take care of the genetic material of the virus, and this retrovirus means the genetic material of this virus is a RNA. So it contains HIV contains a single stranded RNA, and this it and along with this it contains three structural genes. Three structural genes are encapsulated viral proteins, that is shown in the uh, brown color balls, and another one is reverse transcriptase that is essential to convert this RNA into DNA so that it can be integrated into the host genome, and another one is integrase that helps the translated protein that is formed within the cell to form a uh, functional structure. And so these are the important three structural genes that are present within the genetic material of HIV. And in this second figure shows how this virus infected and what are the various strategies that are being used to prevent the infection of this HIV. The first one is the fusion of this HIV virus to the cell membrane, and this is done to those cells that have CCR4 receptor and CD4 receptor and CCR5 co-receptor. And one of the strategies to prevent the infection is to have something called fusion inhibitors that prevent this fusion of this HIV virus to the cell membrane. And afterwards, after the cell gets infected, what happens is the genetic material get released, and this RNA is converted into DNA with the help of reverse transcriptase. So another strategy that can be target is to make this transcriptase inhibitor, or inhibit this reverse transcriptase, so that this RNA material cannot be converted into DNA. So the final host integration will not occur, and this virus will die soon, as it will not be a stable transfection in this case. And so what happens after this DNA has been formed from RNA? This DNA then goes to the nucleus, where it integrates with the host uh, genome, and then uh, takes advantage of the cellular machinery to translate the genetic material, and then the integrase helps in forming this HIV virus into a functional structure. So the next strategy that people can target is the integrase inhibitor, so that the integrase will not be able to perform its function. So even though the genetic material is integrated with the host and it is being continuously translated, it is difficult for that material to do to infect other cells, or to do much harms to the cell, as it is non-functional in this form if integrase is inhibited. So, so after this so suppose the integrase inhibitor is not there in normal process, what happens is after that normal infection process, what happens is after the integrase of this translated protein, it will then form into a functional virus structure that can be uh, that can move out of the cell through exocytosis and then affect other cells. So in this case, what can be done is to introduce some of the to use protease inhibitor. So what is protease inhibitor does is they will prevent the binding of this new exocytosis, new virus particles that are come out from the cells by exocytosis to affect another cell by covering this GP20, or uh, not by covering this GP20. The protease inhibitor does is it prevents the further functional transfection, further functional infection of this new viral protein into another cells by inhibiting the protease. So what happens in case the protease inhibitor works is that it will not be able to it will not be able to it will not be able to do this function in when it is infected into the new cell, that is after the genetic material is translated. It needs to be cut into specific it need to be cut into specific at specific sites, and that is usually done by protease inhibitor. So if this protease inhibitor that is usually done by protease. So when this protease inhibitor is there, so the translated product will not be able to form into a functional structure as in the case of integrase. So these are the few strategies that are being used that can be used to prevent the HIV infection. This case is valid when the person is at the initial stage or not get infected. And this C portion shows how the HIV virus has modified itself to prevent it from the some of the developed strategies, that is TRIM5 inhibition by TRIM5 alpha. What this TRIM5 alpha does is, as we have discussed is in one of the previous videos of HIV virus. Let us quickly go through this in this video also. This TRIM5 does is prevent, as soon as cells gets infected by the virus, it prevents the further translation from RNA to DNA by affecting the reverse transcriptase. This will prevent the reverse or it will affect the function of reverse transcriptase that comes with the viral infection. So how this HIV has modified itself is that this is the usual case that has been shown, that when the cell gets infected with the virus, the TRIM alpha prevents the virus from converting from RNA to DNA by affecting the function of reverse transcriptase. However, what happens in the modified HIV virus is that when the virus gets into the cell, and if the reverse transcriptase gets affected, then what it will does, it will form a different mutation within the nuclear membrane or within the host genome, that is change the G to A mutation, which is the hyper mutated HIV genome, which converts the host genome into hyper mutated HIV-1 genome, and then this results in affecting the normal function of the cells. Thus the TRIM5 alpha will not be able to perform its function in the more advanced HIV virus. So this is this slide shows how the virus progresses inside a body. So this red line shows that initially when the virus people get infected with the HIV, the symptoms are really high, because at that time antibodies against the HIV within the body is very low, and also the immune cell is not trained enough to take care of this new virus, take care of these new foreign things that are that are attacking the body. So initially the acute response is high, symptoms are very high. After that, symptoms gets down because the antibodies against HIV increased, and also the immune system starts to learn about this kind of virus and perform its function in an efficient manner. And after this time, this tagline and this yeah, after this tagline, what happens is there is a asymptomatic state where people does not get any symptoms, but the virus is slowly progressing within the body. And what happens is during this point of time is our body get adjusted to this new virus, so our body stops responding or to this HIV virus because it starts considering it as a part of the body. And also the HIV antibodies will not be, because of this HIV antibodies uh, will remain stagnant as the immune system is not giving the signal to increase the HIV antibodies. And uh, also we can see that the because the immune system is not responding after this point of time, slowly the cytotoxic CD8 plus T cells is also start to reduce because the signal from the immune system is stops uh, to give the signal that their HIV is a harmful thing to the body. and and this black dot indicates that when the during the acute stage the transmission risk of transmission is very high because the number of HIV virion plasma uh, plasma is very high at this stage. After that, the risk of transmission is low because the immune system starts working, and the uh, progressive progress of this virus within this time period is very slow. But after this deadline, after this threshold, the risk of transmission increases tremendously because now the virus starts to the viral concentration starts to increase exponentially, as can be seen. And this second line of figure is shown the viral diversity. As we can see that when the virus is in virus, as we know that when the virus affect the body for the first time, the diversity is very low because it starts at a single point or from the single cell. After some time, when the virus spread across the body, it starts to replicate faster because now it will have many cells that are affected and that are replicating. So because of this, viral density after this threshold increases tremendously. And this uh, black line in the second graph indicates the CD4 plus depletion in GULT. So this remains stagnant. This shows that CD4 plus cells remains stagnant within the body. That helps the HIV virus to continue to replicate within the bodies. So those patients that have stable concentration of CD4 plus during this time period or during its life, then they are and at the same time they also need to have CCR5 co-receptor. If both of these things are present within the cells of the host, and they are infected by the HIV virus, then there is a possibility no action has been taken in due course of time, the virus will infect to those patients, though slowly but progressively, and their condition will look similar to this graph. So this graph will I think this graph helps you to understand how this HIV virus progresses and finally takes the form of AIDS. So that's all for this video. Thank you for your time.